All right, in the last lecture I talked to you about external anatomy, and so now we're going to look at the internal anatomy of a fish. And so here's a general diagram, and again, everything that's kind of highlighted are the things that I want you to know. But some of these other things like dorsal fin, let me get my pen up here. We've already mentioned dorsal fin and adipose fin. Vertebrae probably also would be good to know. A lot of this stuff is straightforward and you already know them. A couple things I also want to point out. The upper jaw or the top lip we call the premaxilla, and the bottom is called the dentary, and that's going to become important later. We'll talk about Pyloric Caeca a little bit later. Those are interesting structures that some fish have and some don't. So take a look at this and make sure you can identify them, but we're going to talk about each of these things in a little bit more detail. Here's a figure from Fishes of Tennessee showing again the location of some of these items and things that I'm not asking you to know. For example, the urinary bladder. You should know where that is. It's just in my experience, the bladder is very difficult to see in a fish. And something like a mammal, it's a lot bigger, it's a lot easier to see. But so we don't often ask you to identify the urinary bladder, but you should be able to figure out where it is. First thing I want to talk about in more detail is the gas bladder or the swim bladder. And we have two basic types of swim bladder. We have the Physoclistous and the Physoclistous. And the Physoclistous is attached to the esophagus, and the fish can gulp air. And so it can fill that gas bladder just by gulping air from the surface. The Physoclistous is closed, and so that's one way to remember the difference between them. The clistus is closed, meaning it's not connected to the esophagus. And so if it's not connected to the esophagus, how do you fill it with air? How do you get gas in there to regulate the buoyancy? And this is done through diffusion. And so this is a schematic of a typical Physoclistous gas bladder. And you can see you've got arteries running in, and then you've got a couple of these nets of capillaries called White Myomeres. And these are going to turn up in several different places within a fish, and you can see them in other animals too. That means wonderful net. And so it's a net of very small, very fine capillaries, which is designed for gas exchange. And we'll talk about this in detail, how the fish does this later on. But the fish is able to cause gas to diffuse out of their blood and into the gas bladder. And so you might think of pumping up a gas bladder would require active transport or actively pumping air, and that's not true. It's just simple diffusion. The concentration of gas is higher in the blood than in the gas bladder, and so that causes gas to diffuse into the gas bladder and it inflates. And then they're able to do the reverse if they need to deflate the gas bladder. We'll talk about the details of this later on in the semester. The White Myomeres, as I said, they show up in other places in fish. For example, anywhere where you need gas exchange or heat exchange within the body. And so, for example, fish that live in cold water often have White Myomeres around their eyes or their brain to help basically the heat generated from the body warms up the blood, and then that is used to keep the eyes and brain warm. Sometimes you see them in muscles for exchanging gas. So the White Myomeres are going to turn up quite often. Here's a diagram of the major arteries and veins in the circulatory system. And these are also, can be difficult to see when you're dissecting a fish. I'm not asking you to know any of these, but just note where the blood flows. And so the important thing is you've got the heart up here in the throat, and then that's located very close to the gills, and so the blood flows to the gills where it exchanges gas and other things, and then it moves along this dorsal aorta to the rest of the body, and gets distributed to the rest of the body before making its way back to the heart. Now, this is what we call a single loop circulatory system. And that's different from what humans have. We have a double loop system. The single loop system, the blood goes from the heart to the gills, then to the body. Whereas in humans and other mammals, you have the double loop, where the blood goes from the heart to the lungs, back to the heart, and then to the rest of the body. It doesn't happen that way in fish. Consequently, the blood pressure is a lot lower in fish. The reason that blood comes from our lungs back to the heart is so that it can get another boost of pressure because we're a lot bigger. Fish don't have that. Like I said, the heart is located well up into underneath the throat, where the throat would be. And fish only have a two-chambered heart. They have one atrium and one ventricle. They do have a couple of accessory chambers, which are not really part of the heart, but they help the heart function. These are called the sinus venosus and the bulbous arteriosus. And so sinus venosus, you see vein in there, so that tells you that it's on the low pressure side of the heart. This is where the veins come in. The bulbous arteriosus, you get artery in the word there, so arteries carry blood away from the heart. These just help collect the blood for the heart, and specifically the bulbous arteriosus, one of its functions is to help maintain blood pressure. So we said blood pressure is low in fish because they have a single loop system. The bulbus arteriosus is kind of elastic. It's kind of like a balloon. And so when the ventricle pumps, then that swells up and then it slowly releases that pressure and slowly squeezes out. So it maintains a higher pressure by having this. And the sinus venosus is not easy to see, but the bulbus arteriosus often is easy and shows up. Of course, it's going to be toward the anterior of the fish because the heart's going to be pumping toward the head, which is where the gills are. The heart here is always pretty easy to find. You're usually looking for that ventricle. It's kind of a pyramidal, like heavy muscle though. So a lot of times just by texture, you can figure out where the heart is. And in case you're wondering, in this particular picture, this is a liver, and it's heavily infected with some sort of fluke parasite. And so that's not a typical liver, just in case you were wondering. Excuse me. Once the blood leaves the heart, it goes to the gills, where lots of things occur at the gills. But of course, the gills need to be thin and light so that you can have exchange with the water. Now, the main beam, or the foundation or the structure of the gill, is called the arch, right? And that's this heavy part here. And so that gives it structure, and then that's attached at the bottom and at the top. It's attached to the fish. And then coming anterior off the front of that arch are stiff finger-like projections called rakers. And then coming off posterior are the filaments. And the filaments are where the blood flows and where gas exchange takes place and other things. And so you can see the structure of the gill here. Then if you look at this, this is a top-down cross-section. So this is like you cut through the gill here and then turn it on its side and look down. So you can see those filaments. You've got two filaments that come off to the posterior part of the arch, and then the rakers point anterior. Now, if you look at those filaments, like I said, they're thin, they're delicate. This is really like the most vulnerable part of a fish. So when you're handling fish, you're pulling them out of the water, this is the most likely thing to get damaged. And if their gills get damaged, they're going to bleed heavily, and it's very hard for them to survive. But they have to be delicate, they have to be thin, because you want to exchange gas and other ions with the water. And so you need that very thin tissue. Again, if you look over here on the right, you can see that those filaments coming off, and they're sort of in pairs. There's like two of them coming off. That big part that's coming off is called a primary lamellae. And then on top of each of those things, it's kind of, those things are a little bit shaped like an airplane wing or something. And then on those primary lamellae, you see these secondary lamellae, like half moon shapes that are mounted sort of perpendicular to the primary lamellae. And then what I also want to draw your attention to in this figure is this figure is showing you the direction of blood flow. And so if you look within those secondary lamellae, you see the blood is flowing this way. And that's toward the head, basically. Whereas in this line here, you see the water current here is flowing in the opposite direction. That's called a counter-current mechanism. And when we talk about respiration later, we'll talk about why this counter-current mechanism exists and why it makes respiration much more efficient. So as that blood flows through the secondary lamellae, then you see it collects in these blood vessels that run along the primary lamellae, which takes them back to the arch. And then within the arch, you've got those larger blood vessels that flow in the arch, and then eventually are going to, one comes from the heart and one goes to the body. Here's just another picture of some gills, again, looking at the blood vessels that run through that gill arch, the rakers that point anterior, the filaments that are posterior, and within the filaments you can see the long flat part is the primary lamellae, and then the perpendicular to that are these secondary lamellae. And this picture down here is showing you different types of rakers. And so these are rakers from different fish, and we often use the rakers as a way to identify one species from another. We do a lot of that in the Lipomus, the bluegills and their panfish relatives. We use the rakers to identify them. We can also kind of figure out what the fish eats by looking at these rakers. And so here, this fish, A, has got very narrowly spaced fine rakers. And so it's going to be able to filter very small particles out of the water. Whereas here, this fish has got very short and stubby rakers. It's not going to be able to filter the water well at all. And that's what the rakers are for. They're for filtering water, for trapping food. Mucus collects on there and collects with the food, and then they're able to work that into the throat and swallow it. And so with these fine rakers, you're looking at more of a planktivorous fish that's filtering small things out of the water, whereas a piscivorous fish doesn't necessarily filter the water. It might use these rakers to help hold its prey in the throat. Here's another example of a Planktivore versus a Piscivore. This is a silver carp raker, and it's so fine, it's more like a sponge. It is an extremely small pore size of these rakers, and so they filter out very small algae. Whereas this is probably like a largemouth bass on the right, and you can see stubbier, heavier rakers, which are not so much for filtering, but for holding prey. Here's another diagram showing the location of the gills and the gill arches. And you've got 4 or 5 on either side. Sometimes that 5th arch is modified to form pharyngeal teeth, and we'll talk about those here in a second. They're also showing something that not all fish have, but some fish have a pseudobranch. A pseudobranch is kind of like an embedded gill-like structure. It's from a different origin within the embryonic fish. It's not a true gill, but it does kind of function like a gill, and it's got structure and blood like a gill. So what are these used for? I'm not sure. I've read several different things. Like they're important for osmoregulation. They're important for getting oxygen or maintaining interocular pressure, blood pressure, and you can see that they're associated closely to the eyes. I've read where they might be used to sense oxygen levels in the blood, and so this is how the fish senses whether or not it needs more oxygen. But they're somewhat similar to gills, but they're not gills, and not all fish have them. And again, I mentioned the 5th arch often is modified to form pharyngeal teeth or throat teeth, and I'll show you some examples here in a second. Okay, that was the circulatory system. Let's talk briefly about the excretory system, which consists mostly of the gills, kidneys, and bladder. And so you're probably familiar with the kidneys and the bladder as part of the mammalian excretory system, but the gills are very important, not only for respiration, but also for excretion. And so the gills are multifunctional, and again, that's why they're so critical. They're very delicate, and if they get damaged, it can have wide-ranging effects on the fish. Of course, they're used for gas exchange, and so dump off your carbon dioxide, pick up your oxygen. They're used for osmoregulation, so they can actively pump ions one way or the other, depending upon you're a freshwater fish or a saltwater fish. But to help maintain the saltwater balance within the fish's cells. They're important for acid-base regulation, so adjusting the pH of the blood. That occurs, a lot of that occurs at the gills. And we said they're part of the excretory system. This is where fish get rid of ammonia. And so a large part of your waste is excess nitrogen that you got from digesting protein, right? Protein has a lot of nitrogen. You often don't need all that nitrogen, and so you get rid of it. And there are many different ways. We're going to talk about the excretory system in more detail later, but at the gills, the fish get rid of ammonia through their gills. Now, the kidneys and the bladder function like they do in many other animals, in that they filter the blood, form urine. The bladder collects the urine, and then it's excreted. In fish, they're not well defined, and they're often difficult to find. So the kidneys aren't well-shaped, obvious organs like they are in, say, like a mammal. All right, and so this is a ventral view. So we're looking from the belly up. You can see the spinal column right there. And so these dark patches that lay on the dorsal portion of that cavity, those are the kidneys. And so they don't look like much. They almost look like just like swellings of a blood vessel. Now, if the fish is infected with something, often these kidneys will swell up and they'll be much more obvious. And as I mentioned, the urinary bladder is just very difficult to see in a fish. Okay, let's talk about the digestive system. The most obvious organ that you'll often see in a larger fish when you cut them open is the liver. And so this is one lobe of the liver for this fish. They often have kind of two large lobes, but the liver is going to be attached to the stomach and the intestines, where they all, the stomach and the intestines all come together, and the liver is going to dump its bile in there as well. In preserved specimens, it's not quite as obvious, but again, texture will help you identify the liver because it's a very soft, easily ripped tissue versus like the stomach, which is much more muscular. Now, a lot of fish don't have a stomach. Herbivores often just have a very long coiled intestine, and because they're eating plant material, trying to break up cellulose, they need to leave that food in their body for a long time to give the acids in that time to work on them. And so they just have this very, 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 very long intestine. And so again, if you cut open a fish and you see a long intestine, you can sort of infer what kind of food it eats. Some fish, like the gizzard shad, have a gizzard, which is not really a stomach. It's just more of a very muscular organ that has grit in it, pieces of sand, small pebbles, and that's used for mechanically breaking up the food, but it usually doesn't release any like digestive enzymes or acid or anything like a stomach would. The gall bladder is part of the liver, and so it stores some digestive enzymes and mostly bile, which is produced by the liver, which helps to aids in fat digestion. And so this is associated with the liver, kind of tucked in between the liver and the stomach to store those digestive enzymes. But it's a very delicate little sac. And so when you dissect, a lot of times it's easy to puncture this and you can't see it. Some fish have structures called Pyloric Caeca. And so the pylorus, or the pyloric sphincter, is the connection between the stomach and the intestines. It's the posterior portion of the stomach. And so at that area, you sometimes have Pyloric Caeca, which are little finger-like projections that come off. And so they help to increase surface area. They're also probably important in fat digestion. And so let's look at some pictures of these. And so this is showing you a picture of what a carnivorous fish would look like versus an herbivorous fish. And so in some of the minnows, this will be a way to tell what kind of minnow you've got. You'll cut open its belly and see what kind of digestive tract it has. Usually, especially in fresh specimens, when you're dissecting them, you can see a dark little pouch associated with the intestines, and that's the spleen. And so this produces blood, red blood cells. It helps to filter and store red blood cells, and that's why it's got the dark red or black color to it. And so it's usually pretty obvious. So here's a picture where you can see this very obvious dark little structure associated with the intestines, and that's the spleen. And in this fish, here we have the stomach, which is very large and it looks like it might actually have some food in it. Here's what the gallbladder looks like in a fresh specimen. And so this has got bile in it, and you can see the yellow-colored, straw-colored bile. But you can see that this is a very delicate pouch that's easy to break if you're not careful during your dissection. And in preserved specimens, it's a little bit harder to see because you don't get to see this nice color. This is what Pyloric Caeca look like. And so if you look here, you can see there's tons and tons and tons of little finger-like projections. All right, and this is at the posterior end of the stomach, at the junction of the stomach and the intestines. And you can see how this can increase surface area. Here are some Pyloric Caeca, they're just bigger. This looks like a bunch of bananas, right? Look, there's one, there's one, there's one. So if you take your probe and you kind of tweeze this up, you'll see it's just like a little finger-like projection. Again, here's that fish. This is not a typical liver. This liver is infected with flukes. Here you can see the intestine. So you can see where these are joined and then the intestine comes off from the Pyloric Caeca, or where the Pyloric Caeca attach. Okay, that was digestive system. Let's talk reproductive system. Not a lot to talk about. It's very easy to find in females. I often say that if you're not sure that whether or not you have a female, you don't, because females are very easy to see unless you've got an immature fish that has not yet produced gonads. But it's pretty easy to tell when you've got a female fish. Male testes are more difficult. The testes often look like fat. And so, again, it's a texture thing that you can kind of feel with your tweezers. The testes feel different than fat, but also I'll show you some things to look for. And so here's one ovary, right? You can see all the eggs. This has got a very thin, clear membrane holding those eggs in. Some fish, it's more opaque, but really the ovary is a big sac that holds eggs. Here's an example. Again, it's a big sac that's holding eggs. And so if you cut it open, a lot of times you can tell whether or not it's got eggs in it. Another thing to look for those ovaries is there's always going to be a big artery feeding them. And so this is one ovary. There's another one on the other side. This is the swim bladder right here, in case you're wondering. And so a lot of times I'll look for that big artery. But the testes have those too. But you can usually tell if it's an immature fish or a female that just spawned, you can still tell that this is like just an empty sac for holding eggs. Now here's testes. And so you can see this big, long, yellow, this looks like maybe a sturgeon. And so that does kind of look like fat. One thing to look for is that just like there are two ovaries, there are two lobes to the testes. And again, you see this kind of looks like white fat, but they always come together right at the urogenital vent. And the same is true for the ovaries. They're going to come together where that urogenital vent is because that's where they're going to release the sperm or the eggs. And so if you're trying to figure out if you're looking at a gonad, just kind of trace it posterior or start at the urogenital vent and see what's there and trace it forward. And so you can see here's a lobe and here's a lobe. and they came together right where that urogenital vent was. So that's something to look out for. These will also probably have blood vessels serving them, but maybe not as big as the ovary. Okay, so let's talk muscle a little bit. Fish are mostly white muscle, which is anaerobic, sprinter's muscle. There is some aerobic muscle in fish, and that red muscle is usually a thin strip underneath the lateral line. And then some fish also have pink muscle, which is kind of a mix of these two fibers. And depending upon the activity of the fish is going to influence the relative amount of red versus white fibers. So something like tuna that swims a lot has a lot more aerobic metabolism, and it's got more of a red-pink muscle. Whereas most fish don't swim that much, and so they just have that fast twitch muscle, or that white muscle. When you eat a fish fillet, most of the time you're eating white muscle. That fillet, those large lateral muscles, are called myomeres. And so just like, you've got a bicep and a tricep, and your muscles have names, these are named myomeres. And so if you look at the side of this catfish here, you can see all these muscles, all these striations. Those are all individual muscles called myomeres. In some species, we count the myomeres, like the postanal myomeres in lampreys, and we use that to help us identify different fish. Here's a kind of a three-dimensional view of what those myomeres look like. So here we've removed a bunch of the myomeres, and there's just 3 of them. And you see it's got this very complicated sort of W shape that's attached to the spinal column and also to different parts of the fish. And so when these muscles contract, it's a very complicated movement. And so when you think about the movements of fish and how what they swim and the different kind of swimming movements that they have, it's because they have these oddly shaped muscles. It's not just like a straight muscle that you'd find, like in your arm or something. And so here's a couple of fillets. And so this is the inside of the fish. And so this is the part of the muscle that's up against the internal organs. And you can see it's all white muscle. This is the fillet from the other side of that fish. And this is the exterior, this top one. Now you're looking at the exterior of the other fillet, and you can see that red meat. And here's where the lateral line went. And so you can see that red muscle that's just underneath the skin. And so that red muscle, because it's got a lot of myoglobin in it, it's aerobic muscle, and so it needs to carry oxygen. And that myoglobin and things that carry oxygen often have a strong flavor. So usually when people are eating fish, they're going to clean and they're going to cut away that red muscle because it tends to give an off flavor. Unless you get something like a tuna or something where it's got a very unique flavor and that's what you're looking for. Okay, the brain skull area is called the neurocranium. And what I want to focus on is the neurocranium. I mean, you're aware of the skull and the brain. That's pretty straightforward. But just posterior to the skull is a kind of an outgrowth of the skull, or a bony structure called the otic chamber. And in the otic chamber, there are 3 pairs of otoliths. And so this is part of the inner ear. In fact, you have small otoliths, but they're usually bigger in a lot of species. The largest otolith usually is called the sagittal otolith. And so in lab, when we start removing otoliths, we'll be looking for the sagittal otolith. And just like your inner ear, the fish inner ear is used for balance and hearing, and the otoliths are an important part of that. We'll talk about this in more detail when we get to mechanoreception later in the semester. Humans also can use the otolith to age the fish. They're a very reliable structure in many species to tell how old the fish is. You remember we talked about how you can use scales to age a fish. The otolith in a lot of species is way better than scales. So this is a top-down view, a dorsal view of the skull, and you can see the brain, and then posterior to that you can see this otic chamber. And the 3 types of otoliths are the sagitta, astericus, and the lapillus. And like I said, the sagitta is usually the largest. Here's another look at one side of the otic chamber. You can see it's got, here's again your sagittal otolith. Inside there your asparagus inside there, and your lapillus inside there. And you got these semicircular canals that are sort of in different planes. And so this is what's helping the fish with orientation and balance, because you've got these canals that are in different planes. And inside this otic chamber is a thick fluid called endolymph and some little hairs. And again, when we talk about mechanoreception, we'll get to details exactly how this helps the fish hear and maintain balance. But it's very similar to your ear, and that's because you inherited your ear from our common ancestor with fish. This is what a sagittal otolith looks like when you remove it from the otic chamber. It's a big, heavy calcium protein matrix, and this one is cracked in half, and you can kind of see rings here where they cracked it in half. This is what it looks like under a microscope, and so you can clearly see the rings. 1, 2, 3, and then one here on the edge, 4. So that's a 4-year-old fish. And so these are very clear, and like I said, in a lot of species, this is the best way to age the fish. Now, talking about ears, in some species, the gas bladder is used to help amplify sound, because it's a big empty void that is going to vibrate like a balloon would vibrate when it gets hit with sound waves. And because it's so large, it can, it gets hit by, it absorbs more energy from that sound wave. And so it's able to take more of that energy from the sound wave. And then if it transmits that energy to the inner ear, that can allow the fish to have more sensitive hearing. And so a couple ways that some species do this. Not all species do this, but some species, the gas bladder is very near the inner ear. So for example, in cod and drum, the gas bladder just sits near this otic chamber. And so when the gas bladder vibrates due to sound, those vibrations are transferred directly to the otic chamber. That gives them more sensitive hearing. In some fish, there's a modified vertebrae called a Weberian Apparatus, or a Weberian ossicle. And this is a bone, it's a vertebrae that's been modified to actually connect the swim bladder to the inner ear. And this is found in fish that are known as otophysian fishes. So catfish have this, minnows have this, suckers have this. And so you can see, if you look at this diagram here, you can see vertebrae, right? And you can see the gas bladder here. And you see this bone right here that sort of rests on that gas bladder. And so when the gas bladder vibrates, those vibrations are transferred through this bone up to the otic chamber. Here's a top-down version of the same thing. So here you can see that otic chamber. See the semicircular canals that I talked to you about? And so here are your vertebrae again. And in the vertebrae, in the rib cage, you see the gas bladder. And then here you see that modified bone, that modified vertebrae that touches both the gas bladder and this bone that touches the inner ear. And so that helps to transfer vibrations from the gas bladder to the inner ear, gives the fish more acute hearing. Okay, earlier I mentioned that fifth gill arch sometimes is modified to form throat teeth, and these are called pharyngeal teeth. And many species have these. In several species, for example, in the minnows, we use the pharyngeal teeth as a way to tell, to identify different species, so it's part of the key. These teeth have many uses. A lot of times they're used to crush food, or they're used to just actually help hold on to prey. So, I mean, they're really cool, right? Imagine if you had teeth in your throat. That would be kind of cool. So here we're looking down the throat of a fish, and you can see pharyngeal teeth down here in the throat. Here are the gill arches, and then there's some pharyngeal teeth in this example on top, and you could have either one. Here's just what some pharyngeal teeth look like when they've been dissected out. And so you can see the similarity to a gill arch, right? You can see you've got kind of like a gill arch shaped portion that attaches at the top and bottom. And of course, you're familiar with the rakers that grow off the arch. Well, if those rakers maybe changed shape and got a little harder and you got rid of the gills, you can sort of see how that fifth gill arch can turn into these pharyngeal teeth. Again, this is just showing you how that gill arch can be modified to form those tooth plates. Here's more examples of what some of these would look like. And so we will practice in lab. We'll take some minnows and we'll practice dissecting out the pharyngeal teeth so that we can use them for identification. Here's pharyngeal teeth that I removed from a grass carp. And this is very useful for identifying grass carp because I don't know of any other species that has pharyngeal teeth like this. Look at these little grooves, right? They've got these little grooves in there. And a grass carp eats vegetation, so presumably these little grooves act like scissors. The little grooves kind of run, in between each other and helps to cut up that vegetation. I assume that's what they do. Here's pharyngeal teeth from a freshwater drum. Freshwater drum eat a lot of mollusks. And so they need to, snails mostly, so they need to crush up those shells. And so they've got these big molar-like teeth that are good for crushing shells. Here, this is a picture I took looking down the throat of a freshwater drum. And here you can see what we just saw, the ventral teeth, and then you see some on the top portion of the esophagus. And so it's very much like your molars, right? They can grind against each other and crush things up, but it's down in their throat. Pretty cool. Here's an example looking at some other teeth that you might run into in the mouth. And a lot of these are used to help identify fish, especially in things like, I want to say mad toms, we might use these. And so I've asked you to remember at least a few of the more common ones that we're going to use in the key. Some of these are easy for you to remember. For example, the premaxillary and the dentary teeth. Well, if you remember, premaxillary is the upper lip bone and the dentary is the lower lip bone, then you can remember the names of those teeth. Up here on the roof of the mouth, you've got the vomer teeth, which is a single patch, and the palatine teeth, which are in pairs. And so one way you can remember that is palatine is in pairs. And you also see down here on the tongue, we look for tongue teeth in a lot of species. Kentucky bass and warmouth. We look to see if there's teeth on the tongue, and those are technically called glossohyal. Glossa means tongue. If anytime you see the word glossa, think tongue. All right, now let's take a look at the skeleton. And you're familiar with the typical fish skeleton, what it looks like. We're not going to learn all the bones, but I'm going to highlight some of the more important ones. The hemal versus neural spines, pterygiophores, pectoral and pelvic girdles, epineurals and epipleurals, and then the hypural plates. So let's look at each of these. So your typical fish skeleton makes you think of a comb. And these little spines that come off the vertebrae and point up, or the dorsal spines, are called neural spines. And then on the posterior portion of the fish, you've got spines that point down. Those are called hemal spines. And where you don't have hemal spines, instead, up here on the anterior portion, you have ribs, which are also called pleural ribs. You got pleural ribs and you got epipleural ribs. All right. So one way to try to remember this is neural point north. So the neural ones are the ones that point up. The hemal spines are the ones that point down. Here's looking at a typical vertebrae head on. So this is kind of a cross section. And again, you can see this is a vertebrae that's toward the posterior portion of the fish, and the neural spine is on top, and the hemal spine is on bottom. This other letter A is a vertebrae that's closer to the head of the fish, and it also has a neural spine, but instead of a hemal spine, it's got the rib coming off of it. And of course here in the middle, you've got this hollow portion that the spinal cord runs through. Okay, pterygophores are small bones that are associated with the dorsal fin. And so you've got these small bones that are connected to each other by tiny ligaments. And then those in turn are connected to the rays in the fish. And so this is what the fish uses to extend and retract its dorsal fin. And so by contracting on muscles, it can pull on these and they act like a lever, which is going to take those rays and pull that dorsal fin up, or you can relax and let the dorsal fin come back down. And so that's what we're looking at here. You zoom in on this fin, you can see these pterygophores that are embedded in the muscle and then they're attached to the fin rays. Now these are also important if you're working with fish. A lot of times we tag fish by putting little tags in their dorsal surface, and those tags have a little T-bar anchor, and we try to get that T-bar in between these pterygophores. And so you sort of aim for where the pterygophores would be and get that T-bar on one side of them, and it helps to hold that tag in. Here again is showing you the pterygophores in this drawing of a fish skeleton. The pectoral and the pelvic girdles are just what you think. You think of your clavicle, think of your pelvis. They're homologous to those. They support the pectoral fins and the pelvic fins. The important thing is that if you're trying to dissect a fish and kind of cut up through there, a lot of times you'll hit certainly that pelvic girdle, and you sometimes have to cut through it. It can be a little bit heavy. The epineurals and epipleurals are also called intermuscular bones. More commonly, these things are called floating bones or pin bones, and they're found in species like pike, and also the silver carp and the bighead carp are famous for having these. These are thin little bones in the meat, and these make eating fish difficult. A lot of people don't like eating fish because they don't like the bones. And when they're talking about they don't like the bones, they're talking about these. These are bones that are intermuscular. They're not attached to the rest of the skeleton. They've evolved several different ways and for several different reasons, but they are attached via ligaments to certain, I guess they are attached to some bones via ligaments, but they're very thin and they're just embedded in the muscle. And so if you're trying to take a fillet off and eat that muscle, these bones often stick around. So removing these bones is often something you need to do to get people to eat the fish. You can see their unique shape. They're kind of wishbone shaped. They've got this kind of Y, which if you look down here, you can see, the Y with the long part pointing toward the posterior end of the fish. And so, again, epineural, think the neural spine. So epi is around, and so these are around the neural spine. These are the ones that are at the dorsal side of the fish. And epipleural, think of the pleural rib. This is down at the ventral surface, and these are the ones that are down at the bottom of the fish. Okay, last thing we'll talk about are the Hypural Plates. I mentioned these earlier when we talked external anatomy. If you're trying to get the standard length of a fish, you measure from the snout to the end of the Hypural Plates. And if you've got a live fish, you can just kind of bend the tail, and you can see where it doesn't bend. It doesn't bend because there's a skeleton there. There's a Hypural Plate. That's where you would measure the standard length. So these Hypural Plates are what attaches the caudal fin to the vertebrae. And they're flat plates. And so you can imagine that if here's the end of them right here. And so if you had the fish and you kind of bent it, it would sort of bend around this corner. That's where you would measure the standard length. Here's another look at Hypural Plates. And that's what they look like, and that's where you find them. Okay. One last look at the axial skeleton. Just again, highlighting things that I'd like for you to know. And there's really nothing added here. This is just some of the bones that we looked at earlier. So there's a lot of things to know. We've already looked at some of those when we dissected fish. We're going to be dissecting a lot more fish. So make sure you understand all these. Let me know if you got any questions. That's all I have for internal fish anatomy. So awesome. I'll see you later.